Good afternoon to my doctor and my friends. Today I'm here to present our paper which is quantification of SARS-CoV-2 by microarray technology. This is table of contents. The first is principle of a microarray technology. First is sample preparation and labeling, then the aggregation, then washing off, and finally is image acquisition and data analysis. The principle behind microarray is that complementary sequences will bind to each other. The unknown DNA molecules are cut into fragments by restriction endonucleases. Fluorescent markers are attached to these DNA fragments. These are then allowed to react with probes of DNA chip. Then the target DNA fragment along with complementary sequence bind to the DNA probes. The remaining DNA fragments are washed away. The target DNA pieces can be identified by the fluorescent emission by a passing laser beam. A computer is used to record the pattern of fluorescent emission and DNA identification. This technique of employing DNA chip is very rapid, besides being sensitive and specific for the identification of several DNA fragments simultaneously. The next is method involved in microarray technology. Currently, much of diagnostic testing technology for SARS-CoV-2 is based on real-time polymerase chain detection, which can call as RT-PCR, which detects viral RNA in a sample. Major limitation of RT-PCR diagnostic method are specificity and inability to distinguish between multiple pathogens. Pathogen DX novel detects XRV technology combines RT-PCR with powerful DNA microarray technology to improve specificity and creates new possibilities for multiplex testing. For COVID-19 testing, RT-PCR is most widely used to detect viral RNA in a sample obtained via nasopharyngeal swabs or nasopharyngeal aspirate. The RNA is extracted, reverse transcribed, amplified, and the cDNA strand is labeled with fluorescent dye for detection. This technology creates a new possibility for future respiratory testing. In combining RT-PCR and DNA microarray technology, Detect XRV enables multiplex diagnostic testing, where traditional RT-PCR methods are confined to the identification of single viral pathogen. As a result, a single test would be sufficient to ascertain whether a sample is positive for any number of respiratory viruses, such as SARS-CoV-1, SARS-CoV-2, or various influenza strains. That's all for me.
You have probably known the principles and methods in this microarray technique from the previous slide. Now, let me continue with the advantages and limitations of microarray technique. As it widely known, each technique definitely has their own advantages and disadvantages, including this microarray technique. Let us begin with advantages. First and foremost, microarray technique has high sensitivity and low sample consumption. This is because several molecules can be detected simultaneously when using a low sample volume. Next, microarray techniques are able to perform multiplex detection due to rapid method sample analysis and handling large samples. In addition, microarray techniques can also be easily customized. This customization makes microarray well suited to serve a wide range of research interests for different purposes. And that summarizes some of the advantages of microarray technique. Moving on to the limitations of microarray technique. Firstly, this technique is limited to access and cost. In a simpler word, microarray technique is expensive. A single experiment may cost hundreds or even thousands depending on how many replicates need to be done. Not only that, a large number of probe designs are also needed based on the sequences of low specificity. Other than that, the results of microarray are often too complex to interpret. This is because the data collected will be huge and need a lot of time to analyze. Next, the disadvantages of these microarray techniques is that it only identifies sequences that the array was supposed to detect. In other words, each macroarray can only provide information about the genes that are included on the array. If the hybridization solution contains RNA or DNA species for which there is no complementary sequence on the array, these species will not be recognized. And lastly, microarray techniques have numerous error-prone steps. Thus, the experiment needs to be replicated in order to eliminate sources of error. Now we've already come to the end, and I hope that our presentation really helps you to understand more regarding this topic. That's all from our group. With that, and thank you.